Statins are normally prescribed to lower cholesterol, but a recent study suggests that certain forms of breast cancer may be responsive to statin treatment. Led by Prives of New York's Columbia University, the international team found that when they treated breast cancer cells carrying a mutant p53 gene with statins, the cells ceased propagating in the chaotic manner typical of tumors, and in certain cases even died. However, researchers caution that much more work needs to be done before these lab results can be turned into clinical success. P53 is a tumor suppressor gene known to help control many aspects of cell growth, such as keeping it organized and blocking uncontrolled growth. Over 50% of us have a mutated form of P53. Most of these mutations don't just disrupt the gene's normal job, they also give it the ability to promote, instead of suppress, the development of cancer. To investigate mutant forms of P53 further, the researchers studied cancer cells growing in an artificial system that replicates the three-dimensional structure of the human breast. There they observed cells with mutant P53 genes growing in a chaotic and invasive manner, exactly like breast cancer in the human body. The next phase of the study involved author William Fried Pastor from Columbia University. He and his fellow researchers found that a pathway known as the mevalonate pathway was responsible for structural changes in the disorganized growth of the mutated P53 carrying cells. This pathway is the same one targeted by cholesterol-lowering statins, because it also makes cholesterol. Finally, researchers examined breast cancer tissue taken from tumors in human patients and discovered that mutations in P53 and an increased rate of activity in the mevalonate pathway tend to coincide in humans as well. The authors conclude that these findings implicate the mevalonate as a therapeutic target for tumors bearing mutations in P53.